Welcome to Lecture 7.2, in which we will go over the light-dependent reactions, which are sometimes also called the thylakoid reactions. This diagram is a good summary of the light-dependent reactions, and as with the cellular respiration lectures, I will point out the parts that I want you to know for this class. Our learning objectives for this video are to describe where the light-dependent reactions take place in the chloroplast and the overall inputs and outputs, understand the role of photosystems, compare and contrast the electron transport chain in photosynthesis to the one we learned about in cellular respiration, and begin thinking about the relationship between the light-dependent and light-independent reactions. The light-dependent reactions take place in membrane proteins found in the thylakoid membrane, including photosystems, the electron transport chain, and ATP synthase. Light is absorbed and used as a source of energy, and water is used as an input and broken down. Breaking down water produces oxygen and some protons for the proton gradient, and by the end of the process, NADPH and ATP are also produced. We can break the light-dependent reactions down into several steps. First, a photon is absorbed by chlorophyll, which excites one of the electrons in chlorophyll. The electron leaves, and water is split apart to obtain replacement electrons. The electron that left chlorophyll travels through the electron transport chain, and its energy is harvested to pump protons, similar to the electron transport chain in cellular respiration. There is then a second photon absorption and electron excitation, and the electron is used to reduce NADP+ to NADPH. Finally, the proton gradient is used to produce ATP in the process of photophosphorylation. Before going into light-dependent reactions, let's talk a bit about photosystems. Photosystems are thylakoid membrane proteins that contain pigments, such as chlorophyll A. In plants, there are two photosystems. Photosystem 2 is actually the first and main photosystem, while photosystem 1 is the second. They were named by order of discovery, which is why it's a bit strange. What I want you to know about photosystems is that the chlorophyll and other pigments they contain absorb photons. For chlorophyll, the absorption of photons leads to an electron getting excited, and when the electron is excited, it gets separated from chlorophyll. The absorption of a photon by chlorophyll in photosystem 2 is the first step in the light-dependent reactions. Light hits photosystem 2, the chlorophyll inside absorbs a photon, and the electron, represented by the E- in the circle here, gets separated from chlorophyll. Once the electron leaves, it needs to be replaced. This is where water comes in. Water is oxidized and split into oxygen, 2 protons, and 2 electrons. This means that one water molecule can replace electrons for 2 chlorophylls. The oxygen will diffuse out of the cell and out the stomata in the leaf, while the protons will stay in the thylakoid space and contribute to the proton gradient. Notice that this step is similar to the last step of the electron transport chain in cellular respiration, but instead of ending with oxygen being reduced to water, it begins with water being oxidized to oxygen. Meanwhile, the electron that left the chlorophyll in photosystem 2 begins traveling through the electron transport chain. The PQ here stands for plastoquinone, and it gets reduced to PQH2, plastoquinol. These are similar to the ubiquinone and ubiquinol seen in the cellular respiration electron transport chain. Also similar to the cellular respiration electron transport chain is that the electrons get passed through cytochrome. Furthermore, as the electron travels through the chain, it releases energy that allows for protons to be pumped into the thylakoid space, gradually returning to its original lower energy state in the process. The electron eventually makes its way to photosystem 1. Like in photosystem 2, a chlorophyll in photosystem 1 will absorb a photon. The photon will once again excite an electron, which will leave the chlorophyll. The electron that made its way over from photosystem 2 will replace the electron. The electron that left photosystem 1 will now continue traveling and eventually end up at NADP+ reductase. As you may guess from the name of the enzyme, its role is to reduce NADP+ to NADPH, using the electron from photosystem 1. This part occurs on the stroma side of the thylakoid membrane, which is important because that's where NADPH needs to be for the light-independent reactions. Finally, in a similar process to oxidative phosphorylation, the protons flow down their concentration gradient from the thylakoid space to the stroma through ATP synthase. The ATP synthase then phosphorylates ADP to ATP in the stroma. Since this process is dependent on light instead of oxygen, It is called photophosphorylation. The purpose of this slide is just to show some of the similarities and differences between the electron transport chain in cellular respiration and photosynthesis. In both cases, energy from electron transport is used to drive the pumping of protons across the membrane to create a proton gradient. In cellular respiration, the source of the electrons is NADH and FADH2, made in the earlier phases before the electron transport chain. In photosynthesis, the source of electrons is chlorophyll, and the electrons are ultimately excited by light. In cellular respiration, the protons are pumped from the matrix into the intermembrane space, while in photosynthesis, the protons are pumped from the stroma to the thylakoid space. Both also involve oxygen and water. In cellular respiration, the oxygen is reduced to water, which also takes up 2 protons and further decreases the concentration of protons in the matrix. In photosynthesis, the water is instead split and oxidized to make oxygen as well as protons, further increasing the concentrations of protons in the thylakoid space. Both use ATP synthase to produce ATP using the proton gradient. In cellular respiration, the protons travel down their concentration gradient from the intermembrane space to the matrix, and the process is called oxidative phosphorylation due to oxygen being necessary for the process. In photosynthesis, the protons travel down their concentration gradient from the thylakoid space to the stroma, and the process is called photophosphorylation due to light being necessary for the process. In this video, we learned that the light-dependent reactions occur in the thylakoid membrane. The reactions begin when a photon of light is absorbed by chlorophyll in photosystem 2. This excites the electron, which separates from chlorophyll. Electrons are taken from water to replace the electron in chlorophyll, and in the process, the water is split into protons and oxygen. The electron from chlorophyll travels through the electron transport chain, and the energy it produces is used to pump protons into the thylakoid space. The chlorophyll in photosystem 1 also absorbs a photon, and the electron that gets excited also leaves. The electron that came from photosystem 2 replaces this electron, and the electron from photosystem 1 goes on to reduce NADPH. The proton gradient that was produced by the electron transport chain is used by ATP synthase to produce ATP by photophosphorylation. Overall, there are many similarities to the electron transport chain in cellular respiration. In the next video, we will talk about the light-independent reactions, which will use the NADPH and ATP produced by the light-dependent reactions. Thanks for watching.